SWEDEPAD, Martin Falkenberg, please.
Hello, everybody. Professor Greenhalgh, thanks for this invitation. And dear colleagues, this presentation is on behalf of all the investigators in the SWEDEPAD trial who are doing a tremendous job. There you go. So we've been taught that if you want to do good clinical research, you have to choose between doing a prospective randomized trial or doing registry observational trials. But it is possible to combine these two. If you introduce a randomization feature into a pre-existing registry, you get the registry-based randomized trial. And that can have certain advantages. Such a trial potentially is faster and much cheaper to conduct compared to other randomized trials. It allows large trials, large enough to address essential clinical endpoints, while still having control not only of the randomized patients, but also of all those patients who, for some reason, were not included and randomized in the trial, making results applicable to the general population, while still eliminating confounders due to the randomization. So we wanted to investigate if drug-eluting technology that was primarily developed in and lately embraced totally in cardiac intervention also produces superior results in peripheral arteries. Drug-eluting technology is certainly very promising, but the evidence that we have in peripheral vessels is still remaining, depending on small trials underpowered to address important patient-centered endpoints. They are device-specific, they are industry-sponsored, they almost exclusively address claudicants, and they address surrogate endpoints. So we thought there was a need for a large and independent randomized trial. A few words about the SWEDEPAD registry. It has been around for a long time. It's been online since 2003. All vascular centers in Sweden participate, and each year there is around 3,000 peripheral procedures registered. So in order to address proper endpoints, we decided to make it into two trials. SWEDEPAD 1 addressing CLI patients and SWEDEPAD 2 claudicants. Primary endpoint for CLI was amputation rate, and for claudicants, quality of life, a questionnaire, VascuCall 6. So we calculated the number of patients needed to either prove or to reject a difference in these endpoints, and we came up with 2,400 CLI patients and 1,333 claudicants. So this is an overview of the design of the trial. It's very pragmatic. Most patients are eligible to one of the two trials, and randomization is done during the procedure when a guide wire has been established across the lesion, and it's between using drug-eluting technologies or not using drug-eluting technology. And all devices with CE marking for this indication are allowed. Follow-up is done through registries mainly. We were lucky to receive significant funding, enough to do the trials, and from the government sources. And in addition to that, we have established collaboration with all companies who are selling drug-eluting devices in Sweden, with a price reduction of about 250 euros per device used in the trial. But it's important to point out that none of these companies were involved in writing the trial protocol, and none of them have any access to trial data.
Martin, I'm going to be very, very rude. I'm very sorry, but we want to know why you decided to stop using Paclitaxel.
Coming there.
Okay.
Sorry.
We've got a couple of minutes to tell us.
This is the inclusion rate during the last 3 years. There are twice as many patients included in the CLI trial, and that corresponds to what we know in the registry. We have included around 60 new patients each month until December last year. This shows how the patients look like. In both trials, most interventions are done in the FemPop segment, and 30% are stented. This is the distribution of the devices used in the trial, and it represents market shares in the participating centers, and it also represents when each company decided to join the collaboration. This is the grand total of included patients so far, 2,290 combined in both trials, and that's 2/3 down the road for the target of 3,333. And the reason why we stopped the trial was the publication of Cazzano's meta-analysis that was very surprising to us, and we thought it should be taken seriously. That was 4 months ago, and we're still there. We're still trying to figure out if to proceed similar to in the Basel trial. And my last words would be that it would be an awful thing if drug-eluting technology, of course, causes death in these patients. But it would also be an awful thing if this technology, if proven clinically superior, is denied to patients due to a safety warning if this warning is eventually proven unsubstantiated. And I think that the only way that we can address these issues, investigating new technologies, is by doing large and randomized independent trials. Thank you.
Thank you very much, Martin. Thank you.